Welcome to the latest installment of the AST AJT Journal Club series. Today's Journal Club features the article, Recipient Sex and Estradiol Levels Affect Transplant Outcomes in Age-Specific Fashion, and is hosted jointly by the AST Women's Health Community of Practice and the AST Community of Transplant Scientists. In a moment, I will turn the session over to our moderators, Dr. Amandeep Bajwa from the University of Tennessee Health Science Center and Dr. Dianne McKay from the Scripps Clinic in San Diego. But first, before we get into the main presentation, we have a few housekeeping notes to help you engage with today's session. There's currently a viewership polling question displayed for the audience. Please take a moment to answer this question while we finish the remaining announcements. This Journal Club is being recorded, and the archive will be available on the MyAST website and the MyAST YouTube page 2 to 3 business days after the live session. Please note that your lines have all been muted so that only the presenters can be heard clearly for the archived recording. If you have a question for our panelists during the Journal Club, we encourage you to participate by using the Q&A section of your Zoom webinar panel. Questions submitted via chat may be missed during the live presentation. If there are questions we do not have time for, we will either answer them individually offline, or we will post the full answer and question to the website following the Journal Club. Finally, when you log off at the conclusion of today's Journal Club, you will see a link to a short evaluation survey to complete. Please fill out the survey to help us keep our content current and engaging. I will now turn the session over to our moderator, Dr. Dianne McKay, to begin the presentation. Dr. McKay.
Yes, hi, welcome everybody. And we have a really exciting Journal Club today that's going to be discussing an extremely important topic, that of recipient sex and hormone levels on transplant outcomes in an age-specific fashion. And I'm privileged to moderate this session with my co-host, Dr. Amandeep Bajwa. And we have the privilege of hearing from Dr. Jasper Iske, Dr. Friederike Martin, and Dr. Stefan Tullius to tell us about this important session. As was already mentioned by Brian, this is a session that is jointly hosted by the AST Community of Transplant Scientists, the COTS, and the Women's Health Community of Practice, WHCOP. So with that, I'd like to turn the session over to Dr. Iske. Thank you.
Yeah, hello, welcome everyone. Thank you for joining to our presentation on our latest manuscript. And today we'd like to give you a more in-depth view and discuss our most recent research findings. And actually the presentation will be held by Dr. Friederike Martin and myself, and Dr. Stefan Tullius would like to maybe give a short introduction to the overall AST.
Thank you, Jasper. I think for us, this is very exciting to have the opportunity to present our data. As a research group, it's always fantastic to see that the work is getting recognition. So we are delighted that this work was published in the American Journal of Transplantation, that it had an editorial with it, and that we have the opportunity to present those data here at the Journal Club. I want to briefly introduce Jasper and Friederike. Jasper is a graduate student. I constantly look up on how many publications Jasper has, because they increase by the hour. He has, from a year of working with us, 10 PubMed listed publications, and still is working on more. So he has been fantastic. Jasper is currently in Switzerland, in Zurich, and plans to be a cardiac transplant surgeon. And Friederike is a resident at the Charité in Berlin. Friederike has been working with us in the past, both remotely in Berlin, in Boston as well. She recently received the Women in Transplantation Fellowship Award, and this work is really at the heart of what she wants to do in her research career. I also want to mention two of the authors that have contributed to this work. The first author, Ryuichi Menosono, who has really worked during his fellowship here in the lab on this, and he is now back in Japan, Osaka, and working as a urology resident. And Edgar Milford here who is one of our senior nephrologists who has been critical in moving our clinical evaluation forward. So with that, I'm looking forward to hear Jasper and Freddie present the data, and I'm looking forward to a very lively discussion. Thanks again for the opportunity.
Yeah, thank you for the introduction. So I want to start with this, at first glance, not very scientific picture. So on the left side, yeah, you can see a man. He seems to like blue and he seems to like soccer. And on the right side, there's a woman who seems to like the color pink and flowers. So, of course, this picture might be quite superficial and stereotypical, but we still know men and women are not equal. So this is not only the case regarding sex-typical preferences for colors or hobbies, but also shows in disparities regarding biological mechanisms and pathologies. So we know, or it's known that, for example, women are or more often develop autoimmune diseases and show more often, for example, with Alzheimer's disease. On the other hand, men are more likely to develop serious infectious diseases, including COVID-19, and also more often develop obstructive coronary heart disease. So looking into transplantation, biological recipient and donor sex also has an influence on transplantation outcome. So for example, it's known that the kidneys from female donors show inferior graft survival compared to kidneys from male donors. So one possible reason in this case might be a graft mismatch. Female kidneys are smaller and have a lower nephron count compared to male kidneys. But there are several other donor and recipient sex-dependent factors which might influence alloimmunity after transplantation. So another aspect might be, for example, that several genes which are important for the immune reaction after transplantation, or for immune reaction at all, are encoded on the X chromosome. So for example, the toll-like receptor genes are encoded on the X chromosome, for example. So what we also know is that sex hormones play a crucial role in the regulation of immune system, or of the immune system. And that and it's possible that different sex hormone levels in male and female recipients of organ transplantation might cause disparities for the outcome after organ transplantation in women and men. So furthermore, we know that sex hormone levels change during aging, especially in women. So young women usually present with high levels of estrogens, whereas old women after menopause have usually quite low levels of estrogens. So this also might be important for the immune reaction after organ transplantation, and led us to some research questions. So first, we asked whether the interaction of sex and age affects allograft outcomes after transplantation, and if age-dependent changes of sex hormone levels affect allograft survival after transplantation. So I just want to give you a few numbers to see why biological sex should be recognized in clinical practice, or why it is important for organ transplantation. So you see here a figure showing the percentage of female and male recipients of organ transplantation in UNAS and Eurotransplant. And we see that men are more likely to receive organ transplantation, and more men are waitlisted for kidney, liver, and heart transplantation. We also know that male donor or deceased donors are, most deceased donors are male, but more women or women are more likely to be living donors. So yeah, just let's go back to the raised questions, whether there's the interaction of recipient sex and age affects allograft outcomes, or the donor sex and age, and if age-dependent changes of sex hormone levels affect allograft survival after transplantation. To answer these questions, we first looked into clinical data. So we analyzed the data of over 400,000 patients aged between 15 and 74 years old who received kidney transplantation between 1987 and 2017. So the data were derived from the Scientific Registry of Transplant Recipients, and we had to exclude 171 patients due to the lack of survival data and 17 patients due to the lack of gender data. We also excluded patients of renal plus non-renal transplants. So what we did with the remaining 407,000 patients was that we divided all these cases in according to their age. So we had one young group with patients aged between 15 and 34 years, a middle-aged group with patients between 35 and 54-year-old patients, and an old group with patients including patients with between 55 and 74 years. So what we analyzed first was whether donor and recipient sex and age affected 5-year death-censored graft survival. And we could observe an inferior graft survival, especially in young female recipients. This was even pronounced in young female recipients who received male kidneys. So especially the combination of a male donor and a young female recipient was associated with inferior graft survival. Most interestingly, we observed that with, no one slide back, please. So we observed that prolonged graft survival in the middle-aged group, or we observed a prolonged graft survival in the middle-aged group independent of the recipient sex. And also very interesting was that we saw an even more prolonged graft survival in the old group, especially in old female recipients, which showed even a more prolonged graft survival compared to their male counterparts. So next we compared young and old female kidney transplant recipients, and we saw a significant reduced graft survival in young compared to old recipients independent of donor sex. So additionally, we calculated the odds ratio for treated rejections within 1 year by recipient sex and age. And what we found was a twofold increase of acute rejection in young female recipients compared to old ones. So furthermore, we wanted to know whether sex by itself does impact graft survival. And our analysis showed that sex by itself did not impact graft survival, but the combination of recipient sex and age has a significant impact on the graft survival after transplantation or after kidney transplantation. So taken together, all our clinical data reinforced our idea that sex and age dependent differences in sex hormone levels could have an influence on graft survival after organ transplantation. And yeah, with this, I want to pass the word to Jasper.
So yeah, I'm back. Hello. So exactly. So like, thanks for the start, Friederike. What we did next was we wanted to delineate the immunological mechanistics behind the observed clinical discrepancies between sex divergent donors. So we set up an in vivo mouse model transplanting male donor skin grafts onto young male and female, as well as old female recipients. Moreover, since female hormonal levels change during the course of aging, as well as the estrous cycle, we performed ovarectomy on recipient animals to deplete or more or less compromise the intrinsic estrogen production. And so when we then measured serum estradiols level, more or less as expected, we observed high levels of estradiol in young female mice that were significantly diminished in old females. And notably, young male animals showed similar estradiol levels as old females and the ones that had undergone ovarectomy. So, consistent with our clinical observations, we could show that young female recipients displayed a significantly compromised graft survival when compared to young males. And notably, and this is like something worth highlighting, is that when we performed the ovarectomy, it prolonged the allograft survival comparable to that of male recipients, which correlates with the observed changes in the estradiol levels. Of note, sham-operated control animals displayed no difference in survival when compared to their young female counterparts. So, to dissect the alloimmune response underlying these observed alterations in graft survival, both clinically as well as in the animal model, we performed a systemic immune response analysis of ovarectomized mice and the sham-operated ones. And we started off with assessing the CD8-CD4 T-cell ratio that is reflective of the cytotoxic immune responses. And we found an increased CD4 to CD8 T-cell ratio in ovarectomized mice, indicating a decreased CD8 T-cell response. And moreover, we monitored frequencies of regulatory T-cells that have been shown to establish immune homeostasis and promote immune tolerance following transplantation. And indeed, ovarectomy increased the frequencies of Tregs, indicating a dampened alloimmune response following the transplantation. So, confirming the hypothesis of an increased alloimmunity in young females was the observation that Th1 and Th17 alloimmune responses, which basically constitute the main drivers of allograft rejection, were significantly dampened in ovarectomized animals, with decreased frequencies of pro-inflammatory IL-17 and interferon gamma producing CD4 T cells. Moreover, the proliferation of CD4 T cells had been decreased through ovarectomy, further supporting the concept of a decreased alloimmune response through ovarectomy. So, as our results indicated that high estradiol levels, or those occurring in young mice, may promote T-cell-derived alloimmune responses following transplantation, we next tested whether increasing estradiol levels may impact CD4 T-cell differentiation and polarization. So, therefore, we isolated CD4 T-cells and stimulated them in vitro in the presence of increasing estradiol levels, reflective of the menopause, the estrous cycle found in young mice, and levels similar to pregnancy. So, and notably, estradiol levels reflective of the estrous cycle found in young mice promoted Th1 differentiation and under Th1 polarization experimental conditions. Moreover, it also augmented Th17 polarization, which is consistent with our results from the immune monitoring following transplantation in the in vivo model. And to be complete of note, estradiol levels had no impact on the in vitro polarization of regulatory T cells. So in order to confirm our hypothesis of an increased Th1 and Th17 immunity due to high estradiol levels, we replaced the deficient intrinsic estradiol production in overreactorized mice through subcutaneously implanted celastic capsules that continuously release estradiol. And therefore we used two different doses, 50 microgram per milliliters, which is reflective of the estro cycle, and a high dose of 500 microgram per milliliter that relates to pregnancy in mice. So, and subsequently we performed skin transplants according to our protocol used before that you've seen in the previous slides, and again performed an immune response analysis and monitored the different responses. So again, we found a significantly decreased frequency of CD4 T cells following high dose of estradiol replacement. And more importantly, the CD8 T cell derived pro-inflammatory interferon gamma production, as well as CD4 T cell derived interferon gamma production, was significantly increased in overreactorized mice undergoing estradiol replacements. You can see here. In addition, Treg frequencies had been significantly diminished, underscoring the pro-inflammatory or pro-alloimmunogenic impact of estradiol levels on T cells found in young mice undergoing the estrus cycle. So ultimately, these observations translated into a decreased allograft survival in estradiol replaced overreactomized mice, confirming the concept of an estradiol derived augmentation of alloimmunity following transplantation. So here you have the overreactomized mice and here the ones replaced. So notably, the estradiol replacement reflective of pregnancy that I just showed up now prolonged allograft survival even beyond that of overreactomized mice, which is consistent with the observation of an improved or augmented immune tolerance during pregnancy. That is important for establishing the fetal-maternal immune interface, which basically allows the semi-allogenic fetus to develop within the mother without being more or less recognized or, let's say, affected by the mother immune system. And at least just to make that clear again, it also proves that estradiol is not promoting alloimmune responses per se, but in a dose-dependent manner that we have just seen when we use the different doses comparing young mice under the estrus cycle, pregnant mice, or even mice that got overreactomized or menopaused mice. So to give you some conclusions, female recipient sex constitutes a significant risk factor, augmenting alloimmune responses and compromising graft survival, which we confirmed both clinically here and in a in vivo mouse model. Recipient age and hormonal levels exhibit a relevant impact on CD4 T cell immunity and graft survival. And at least sex as a biological variable driving alloimmunity and impacting transplant outcome is an understudied area and requires detailed mechanistic studies, just as we've done here in this study, showing that more or less the sex and sex differences in a lot of mice that are usually male in experimental settings actually affects the outcomes that we see in a lot of experiments. So it's basically, it could display a confounding factor for experimental as well as clinical outcomes that needs to be respected. So we thank you all for your attention. And of course, we are now happy to discuss our results, the manuscript, and of course, maybe the clinical setting or any questions that are open. And I'd like to hand over my, my time of speaking to the moderators that will just shortly appear back up on screen.
Okay. Well, thank you very much, Dr. Iske and Dr. Martin, for a really exciting and important discussion. about a really, really important topic, something that we don't spend a lot of time, certainly as clinicians, thinking about. We don't make clinical decisions based on the sex of the donor and the recipient, at least not yet. And maybe that's something that an area that you guys have stimulated some thought on. I have just a couple of questions that I want to direct to each of the speakers. And starting with Dr. Martin, you mentioned toll-like receptors being expressed on the X chromosome. And we know from a lot of experimental data and clinical data that women have less acute kidney injury than men. And certainly when you do experiments in animals, it's very hard to induce acute kidney injury in female mice, for instance. And a lot of this is driven by, we're learning a lot of the acute kidney injury is driven by innate immune receptors, toll-like receptors, intracellular innate immune receptors. And, I just wonder what your thoughts are on how maybe the innate immune system might be modulated and whether this translates into clinical differences that you guys have observed. So is there more delayed graft function, for instance, in female donors that would be due to the acute kidney injury of female donors? I mean, are there any things that we can see? We haven't talked about B cells, and I'll direct that question next to Jasper, but do you see increased donor-specific antibodies? Do you see anything clinically that we can get our hands around as people
So
taking
if I got it right, you want to know if we saw in the clinical part of the study
right.
Differences between the male recipients and the female recipients. So
right, or the donor organs. So you have a donor organ from a female donor versus a male donor put into your combinations. Does that translate into differences in donor-specific antibody responses? Something more granular.
So I'm afraid we don't have these data so far. It was a retrospective study of data. So we know a lot about the recipients and we know donor age and sex, but we have no specific looking into antibodies and stuff. We don't know that so far.
And Dr. Iske, in your studies, have you looked at B cell responses or other?
Yeah, so maybe I can just jump in and point on the aspect that you talked about, the toll-like receptors. Actually, we've done before extensive studies on aging and ischemia reperfusion injury in kidneys, and saw basically that like specific toll-like receptors are crucial for driving the injury there. And what would be highly interesting, we have not done that yet, since there's increasing evidence suggesting that toll-like receptors are, let's say, differently expressed in male and females. And this in combination with the aspect that we've shown differences in the autoimmune response in aged female mice undergoing menopause might be a relevant question to ask to see if females show regarding ischemia reperfusion injury a different response, because we have shown that old kidneys even display an increased ischemic response with higher inflammation. And now what would be interesting is to see if females that are likely to have different toll-like receptor expression patterns react differently to that kind of injury. And then coming back to your second question about the B cells. Well, it's like a topic that is as well is interesting. Our study was focusing on the CD4 T cell compartment, and we have not yet looked into donor-specific antibodies, even though this would be something that might even be of clinical relevance, as this could be tested, let's say, quite easily through experimental setups. And we are, of course, aiming to extend our our experimental studies on not only the CD4 or like CD8 T cell department, but also have a look at innate immune responses and B cell derived immune responses and how these may impact, or even maybe on the other hand, show as well sex-specific differences in allograft rejection responses.
Okay, thank you. Aman, did you?
Thank you. That was a very thorough presentation, and I think we have more questions now than we have answers, but it's a very timely manner, and it was done very well. My question is, you know, historically in preclinical models of transplantation, for minor mismatches, we always take male donors, or, you know, to identify where the cells are coming from and so on. In that context, and historically for transplantation, you know, females, for example, wives can't donate to their husbands because of sensitivity, right? So is this something that we're going to see more of, you know, in the future? That do you think this will change potentially how we allocate organs? Or, you know, I mean, I know right now transplantation is shortage of organs everywhere, so we don't have the luxury to decide who gets it, and you know.
Yeah, I think,
but what are your thoughts on that?
I think that this is one problem, that we don't have the organs to have or to focus on these differences, and I don't think we know enough right now to put it into clinical practice, as we do it with aging. We know that due to aging, allograft reaction or alloimmunity changes, and so there are programs that mention this, or that take age of donor and recipient into account. But I think with sex, we are not far enough with what we know. But I think in the future, if we have more evidence, it might affect, especially in living donation. There, it could be a point to focus on it, and as well on deceased donors, if it's possible. But you're right, that the main problem is the shortage of organs that we have, that we can't focus on that right now, I think.
Yeah. So along the same lines, you know, this is following up on a question that was asked by Dr. McKay. You know, in mouse models of AKI, females are just not susceptible to ischemic injury. So do you think that what you are seeing is a direct effect of estradiol, or it's an indirect effect? So is it something that the hormone itself is driving the change in T cells or immune cells? So what are your thoughts on that, independent of TLR4?
Exactly. So actually the question of like the estradiol impact on T cells is the question that was we had to face this question more often in the past, and the data in the literature are more or less not always straightforward. But what we can say for now is that our data, when we isolated the CD4 T cells, and what we could show is, so basically they were taken out of an in vivo context and were like isolated and put into conditions stimulating the polarization. And that is something that we cannot rule out for now, because we added TH17 or TH1 polarization conditions and estradiol. So the question would be, like, in vivo, where do these polarization conditions do have an environment that provides these cytokines, or even the innate immune cells? So what would be interesting as well is in how far we can take, for example, if we do more or less co-culture experiments with different cell types, let's say dendritic cells or something, subject them as well to estradiol and see how every immune set that may contribute to T cell differentiation may impact this outcome in the end. But what we have to say is, since we isolated them, we are for now at least convinced that there is a straight impact. The only thing that remains open is it an estradiol receptor? Is it like a receptor that takes the hormone and says, all right, go ahead now, change your profile and do something? Or is it the impact of that hormone on intracellular signaling pathways that we have not checked or understood yet so far? So there is data, but it's, to be honest, I would say we're heading along with some pioneers in this group, in this field.
Thank you. Dr. Tullius, do you have anything to add to what Jasper and Friederike already mentioned?
So first of all, I think that the aspect of ischemia and the injury subsequent to ischemia in a sex-specific way is extremely interesting. We, as Freddie said, we haven't looked into the clinical data, but that is something that one could very nicely delineate then also in regard to aging and donor and recipient combinations. In regard to your question, what is the impact of sex hormones on alloimmunity? I think we have shown that there is an impact of estradiol levels on alloimmune responses. At the same time, if you go into the rejection of transplant outcomes in older recipients, there is a difference between female and male recipients there as well, where the estradiol level should not, or we would not expect the estradiol level to be so impactful. So there is certainly more to it, and we have only looked into estradiol and not into any other sex hormones that may play a role, and many other aspects that could potentially have an impact.
All right. If there are any questions, please feel free to type them into the Q&A. Dianne, do you have any other questions?
Yeah, I wanted to follow up on a couple of things. First of all, have you observed any of these differences in your animal models if you use a vascularized allograft model rather than a skin graft model? I noticed in the skin graft models, you went out a couple days. And so I wonder if you might see a more robust difference in a vascularized model, such as a heart transplant. Have you thought about doing any of those types of experiments?
Oh, yes, actually. So in our manuscript, we actually we looked at, we were asked to provide more or less, like you said, like to see if this is in a vascularized setting, can be, if similar effects can be observed in a vascularized transplant model, and therefore we decided to actually what we did to get to a more clinical aspect, as we thought to do the whole thing under CTLR4 Ig immunosuppression. So we used to put this to a clinical context before going to vascularized organs. We used immunosuppression to see, well, can we put this into clinical relevance? Because a lot of, let's say, effects that you see in mice are completely different once you put the animal, or even in humans, use drugs that change the immune response. And what we could show is that these response, like these effects are basically preserved under CTLR4 Ig expression as seen in the manuscript. So you can see that the effects, like the difference, remain the same. So still overreactomy prolongs the survival similar to that of males, and females still remain highly below that survival. And as well, we performed, after that, we performed cardiac transplantation, heterotopic to the, and then we looked at the same survival, and as well there we found like a preserved, let's say, immune response or even graft survival. So what would be interesting now, and this is maybe something linking some of the questions, would be to see B-cell responses and antibody responses that are way more prominent in vascularized transplant models, and to evaluate, like, follow up on this in such a setting, rather than like adhering to the skin transplant model yeah, so this was more, but we've done that. So we've gone through it, and we basically saw these effects preserved throughout all our models, more or less confirming the relevance of it, and even, let's say, the impact. Yeah.
Very nice. We have a question from one of the attendees, and wonder if you can address the issue that both of all three of you maybe. Could you expand upon whether any differences in adherence to immunosuppressive therapy in younger females versus older females versus males could contribute to some of the graft outcomes that you described? Maybe Dr. Martin.
So what we know, so we didn't look into this in our study, but what is known is that young female recipients adhere quite good to immunosuppression, better than the male counterparts. There are differences in with immunosuppression adherence. We don't think that there are differences comparing old and young. That should not be the problem. What is quite interesting, and what the research group of Dr. Tullius examined before is that immunosuppression has different effects depending on age. So, for example, it's known that CTLA-4 for example, works better in younger recipients compared to old ones, and that older recipients don't need need lower tacrolimus doses, for example, to have the same results of immunosuppression. So I think immunosuppression is quite important for the topic of age, and might be important for sex difference as well, and it's something that's under or what we want to investigate further right now. But I don't think that the adherence can explain these differences.
Actually, even like just picking up on that, what you can add here is, if you look at our data in the animal models, which basically the mice do not choose to adhere to any program, they get treated, and we see these effects preserved in our animal models. So what is for sure is that these effects are persistent throughout sex. So the sex is the impact, more or less. What's the only question that could, or maybe to answer the question of the person in the chat, is that it might affect or maybe improve or compromise the effects we have observed, but actually the experimental setting more or less provides the reasonable to say, well, there is an effect, and it's not depending on behavioral features.
So Anita's question, I think, is very relevant. So number one, the most prominent reason for graft loss in the age group between 20 and 30 is non-compliance. And as Freddie said, there is a gender-specific aspect to compliance as well. The interesting scenario that one could envision is, if there is some degree of non-compliance in a particular age group, is there then a sex-specific tolerance or response to that? And that, I think, would be clinically very challenging to investigate, would probably be very challenging to create non-compliance, but experimentally it could be done that one would lower immunosuppression and then see, is there a difference in a sex-specific way to that.
So we know that older people have increased T-cell memory responses, and that might be an area where there may be some differences between older men and women. And so another question sort of going off that, which is really more of a clinical question, is whether you've observed any differences in response to rejection therapy in older versus younger females compared to males. So do older females respond more vigorously or respond better to treatment of rejection, for instance, as younger females? Are there any differences that we can sort out?
To be honest, we again didn't look at this part, but it's quite interesting. So I can't answer this with our data, but that might be a really important thing to look into, if this makes a difference. Yeah, but we don't have the data. I'm sorry.
One other question for Jason. You'd mentioned that you've seen differences in toll-like receptor expression between male and females. I'm assuming these are mice. Can you elaborate on that just a little bit more?
Like, I think it's targeting me, but I have to say, I just picked up on, this is like something you find in the literature. What we did is we found like only to see that age-specific immune responses are mediated through toll-like receptor expression, but not like we haven't looked into we just saw this is the mechanism that contributes to it. And what we saw is that in aging, which might be relevant in old mice and females, is that these these toll-like expressors taken that females show a different expression of them might even show a different age-specific immune response to ischemia reperfusion injury. So this was just my hypothesis taken out of the data that are there yet. But we have like two separate parts. We looked at this age-specific immune response, and I just picked up because we've looked at more or less we identified the toll-like receptor 4 actually to basically mediate an age-specific immune response that is crucial for old organ inferior outcome when transplanted. Yeah, so this is the thing. But actually, I don't know if they are already like, especially which are the ones that are higher expressed in females, and especially what would be interesting is there an organ specific difference in the expression frequency of the toll-like receptors? So let's take the kidney off comparison to the heart.
Okay.
Or even the lung, yeah.
Yeah, sure, very important. We have another question from Dr. Chong, and the question is, what were the estradiol effects in your mouse model limited primarily to T effector cells, but not to DSA responses?
Yeah, exactly. So like we just picked that up, is we have like focused on T cells in this whole experiment and looked at the direct T cell response. So we took this as this is especially in the skin transplant model, basically one of the major driver of allograft rejection. And since we started off with skin transplants, we did not say, okay, let's take the skin transplants and look at the DSA. So the next step was to use a vascularized model that we have established now, saw a persistence of the effects we've seen, but this is not part of this manuscript, but the next step would be to look at DSAs and B cell responses, of course, in a vascularized model, because the skin transplant model is not the let's say, sufficient model for it.
Okay.
So Aman, do you have?
Yeah, I think we don't have any more questions by the attendees. Thank you so much for your time. That was very educational, and I learned a lot. Again, thank you so much to Dr. McKay for co-moderating the session with me. And at this time, we'd like to turn it back over to Brian, and he has some announcements before we sign off.
Thank you very much as well for having us.
Yeah, thanks to both of you for your wonderful moderation and discussion.
Excellent. AST would like to thank all of our panelists and our attendees for today's excellent session. Please remember to complete the evaluation survey and visit myast.org/journal club to view our video archives and register for upcoming journal clubs. To learn more about the AST's community of transplant scientists or the Women's Health Community of Practice, please visit myast.org/COPS or connect directly to the COTS and WHCOP hubs. Thank you all again for today's session.
Thanks.